Callset evaluation is always the ugly duckling of the pipeline. The reason is that this is the part of the pipeline that we really can't automate. Everything that we talked about up to now, including probably half of this slide deck of Callset evaluation, you could write a pipeline that will take your input samples and do all the steps that we talked about, align and mark duplicates and sort and BQSR. And in fact, we have such a pipeline in place already. You can just copy the one that we have. Callset evaluation is different because, first of all, as you will see, you need comparison data. So that means you need to find a comparable set of variants from a comparable set of individuals to which you need to compare your metrics. And then you need to actually sit down and compare your metrics. And the kind of paradox here is that if there exists a call set of samples that are similar to the samples that you're working on, and in a similar environment, similar technology to the thing that you're doing, then what are you doing, right? So you have to be doing something novel enough so that it's interesting, and yet you need to evaluate it with something that is similar enough so that it's not interesting. And so this is the paradox here is exactly why Callset evaluation is more of an art than a science, and it's we have a weekly meeting during which we shout at each other very loudly because we disagree on how to evaluate call sets. And so it's not, I can tell you what we do. I'm not sure I can teach you exactly how to come up with the same answers that we come up with. That doesn't mean that our answers are the correct ones, but it means that there's more art than science here, as I said. Okay, so here we are. We have a VCF. It has all the variants for the samples that we're interested in. We've done all the steps. We've reached the end, and we need to, hand this off to the next person who's going to do some analysis. But before that, we want to make sure that we didn't do any mistakes, that we filtered it at the right place, that there's no, bugs in our pipeline, etc. So, okay. So, basically, we would like to know where on this spectrum our call set lies. Is it, perfectly representing the underlying biological truth? Is it horrible? There's no real variance in it, and there's only artifacts, or is it somewhere in between? Hopefully, it's somewhere in between. Well, hopefully it's here, but it's not going to be here. So hopefully it's here, right? Right. So basically, the premise is we take our call set and we calculate some statistics on the entire call set. Now, before I was talking about statistics that happen at the individual variant level, right? We were talking about statistics that go into VQSR. But this is not what I'm talking about here. Here I'm talking about statistics that take the entire call set and give you a few key numbers that you can kind of compare call sets with, right? So numbers like how many SNPs does it have? How many indels are there in your call set? What is the TVTV? We'll talk about what that means on average in the call set. What is the indel ratio? What is the concordance between this call set and another call set of good quality on maybe a subset of the samples? And then you look at those stats on your call set. You can calculate those stats on a different call set, and you compare them. And then you can say, well, this is something that someone else published. It was good enough for them. Hopefully, my data set is also good enough if the stats are similar. That's basically how it goes. Of course, concordance doesn't have to be, concordance is concordance to some truth set, right? So it's not concordance to some other sequencing project with the same technology. It would be like concordance to some validation data, right? So concordance to something that you really believe is true. It might be a more expensive assay, but it's a high quality assay. All right, so, and here comes the problem. You want to compare your data set to a different data set, but that other data set has to be comparable. So you want to match population ethnicity, you want to match the sequencing and experiment design. So you don't want to compare an exome sequence to a genome sequence. You don't want to compare an exome sequence with one set of baits to an exome sequence with a different set of baits. You don't want to compare a project that is family-based and a project that is not family-based, because the project that is family-based will have the distribution of SNPs and allele frequencies will be very different than the project that is not family-based. Right? That's talking about the experimental design. Cohort size. You don't want to compare your project of 100 samples with the exact project that has 120,000 samples. You will get different statistics there, so you won't be able to compare to that. Some of the statistics are stable to each one of those things, but there isn't any statistic that is stable to all of them. And so we need to make sure that we match so that we can compare statistics. So here's, what is this? Oh, singletons per genome across different populations in the what paper is this from? Some paper, some Nature paper. Different populations, including fins, winning from the bottom, number of singletons. So you can see that, if you were to compare the number of singletons that you have in your call set, well, if you're working with this and you're comparing it to this, then you will think that you have very few singletons and maybe something is wrong. Maybe you are over-filtering, right? So that's just one example. But all of these specifications are going to be different. So it's not easy to do this. And sometimes, as I said, it might be impossible, especially if you're working with a specific population, as many of you might be. And so then you might need to learn which stats are stable with which changes of design, and to say, okay, I'll take my singletons and I'll compare it to a different finished project, even though it has completely different technology, but then I'll take my other stat and I'll compare it to something that's not finished, because I know that that doesn't depend so much on population. So it takes a little bit of work to figure out how you're going to evaluate all the different metrics that you want to use. All right, so here's another example how ethnicity affects some of the variant calling metrics. So this is from the exact data set. There's I don't know which version, this is probably version 1. There's somewhere between, this is the published version, there's probably somewhere between 60,000 and 90,000 individuals in here. Depends whether they were using the data that they were allowed to publish on or not. There's the mix of data that they could use. It's colored by ethnicity, so red is African, because they have more SNPs on average. This is how many SNPs each individual in that call set has after filtering. Okay, so you can see there's lots of dots, one for every individual, but you see that all the Africans are up here, and then you see the Europeans here. This doesn't separate the fins out separately. Maybe these are the fins. This is schizophrenia. It's quite likely that these are the fins, actually. Well, just because there was a schizophrenia study, right? And what else here is interesting? East Asians, Americans, what's that? There are capture batches, but I don't know that we are seeing them here. I don't know what this is sorted by. All right. So why do Africans have more SNPs? African population is an older population. it is a population that did not undergo the large bottleneck of the out of Africa migration. And so there are many different haplotypes in Africa. And when we sequence African population, we see all these different haplotypes, and these haplotypes are in the population. And so you see many of them. And also, of course, the reference was built mainly out of European. And so the reference looks more like this, and so the African looks different. Okay, so what do we use for truth? So it's good for concordance to use an orthogonal method so that you're not validating your method, your lab protocols with your lab protocols. It's not very useful to say that, you know, my experiment from today agrees 100% with my experiment from yesterday, right? Because both of them could be wrong. It's much more powerful to say my experiment from today agrees with somebody else's experiment from a different side of the world on the same people, right? So if you have some control samples that were sequenced or otherwise genotyped on an orthogonal assay, then it's best to use that for your concordance metric. Maybe not for every metric, but for concordance, it would be good to do that. And you can do that on a subset of samples. You don't need to have all of your samples measured against you don't need to have truth data for all of the samples. So the concordance experiment or the concordance assay could be an add-on that you have just to evaluate your call set, and it doesn't have to be as expensive because you can just do a subset of the sample. Other commonly used truth sets, we have to mention dbSNP, even though, as I mentioned at least once before, it has zero curation. Basically, if someone thinks they found a variant, they can put in dbSNP and it's there and that's it. And taking things out of dbSNP is extremely difficult. So it has a lot of false positives in it, a lot of things that are artifacts of mapping and just general poor lab procedures. Sample match genotyping chip, of course, is the best. It adds cost, and if you don't know what variants you're looking for, then you can only use common variants, and so you might not be you won't be able to measure the novel variants as well. You won't know what your concordance is on novel variants because you didn't know to measure them to begin with, right? Because you designed the chip before you found the results. Of course, you could design the chip after you find the results, but that's even more expensive. The HapMap has relatively high rate of validation on common human variants, and so that's a pretty good data set to use for variant concordance. If you also sequence some of the HapMap samples as controls, then and you include their data in your call set, or at least if your cohort, for the purpose of making the call set, you include some of those samples, then you can see how you do on the HapMap samples. Similar with Omni. And NIST, the American National Institute for Standards and Technology, has released a thing called Genomes in a Bottle, which is both a physical sample that you can take and use in your experiment and an associated data set. And they've done this for several samples, including NA1278 and several of the 1,000 genomes samples. So this is very, very common to use this as benchmarking, although that's usually benchmarking for methods development, not so much for assay evaluation. All right, so what are the metrics that we look at? I talked about the number of SNPs and indels. We know what SNPs are, we know what indels are, so that's easy to understand what that means. That can be calculated for the entire cohort, or it can be calculated at a sample level and look at a distribution. Indel ratio is the ratio of insertions to deletion. So basically, how many insertions did I have? How many deletions did I have? And you can just look at that as a number. Genotype concordance, basically you take this other sample, this other data set that you have that's orthogonal, and you ask whether your calls are concordant with the calls that were made there. Hopefully, again, the calls that were made there are things that you trust, a different assay that does not have the same bias as whatever assay that you were using. So if they agree, then you call that a concordance. If they disagree in any way, then it's discordant and you don't count that. And the TITV ratio uses a curious feature of the variant, the true variance in human, which is that so the transitions from A to G, G to A, C to T, T to C happen at a much higher rate than the transversions, which are all the other mutations, even though there are more ways in which you could, if you didn't know anything, you would think that there's more TVs than TIs, right? There's twice as many lines that are TV than TIs. And so even though that is the case, the TI-TV ratio, meaning the TI divided by TV, is higher than 2 in good call sets in humans. So there's a lot more TIs going on, a lot more transitions and transitions. Yes?
Why don't you use that as a prior?
Why don't we use it as a prior for genotypes? I don't know. Yeah, it's a bit of a chicken and egg. It's a good idea, I think, but it would mean that you can no longer use the TITV ratio for evaluation purposes. So maybe as a post a genotype refinement step, but you're correct. I mean, there's the truth is that the difference between this and a flat prior is very small. I mean, I'm saying the transitions are basically 4 times more. Each specific transition is 4 times more likely than each specific transversion, right? So so a ratio of 1 to 4 is maybe what you would get out of this. But if you look at the genotype quantities that we get, we're talking about Phred scale 60, right? So Phred scale 60 is 1 in a million. So how much is that going to be affected by an extra factor of 4? So it will be 62. Okay. I'm not sure that that's what's going to change the quality of your call set. What Laura was presenting before, that was using population priors, can take much bigger priors into account, right? Because you could take variants that are 1 in 1,000 in the population, 1 in 10,000 in the population. So that adds a big chunk to your genotype quality. But 1 in 4 is a really small. It's basically flat. Right. So a number of SNPs and indels. This can be used as a sanity check per sample. These are the numbers that we're expecting. Whole genome around 4.5 million. Exome is about 40, 50,000, something like that. But of course, as we saw before, it really depends on the population. It's not enough to just hand wave, because you could get it really wrong. I mean, I once realized that our pipeline was wrong because there was a 5% error in the number of expected variants, right? So a 5% error could indicate that there's a bug. It's not necessarily just yeah, it's all right, right? So here's the TITV. So these again are expected numbers. For genome, we're expecting something like 2.1 in humans. For exomes, it's higher. It's 3 almost, or higher than 3 can be. If you have low TITVs, that's usually an indication of a large amount of false positives, because the random case has a TITV of 1/2, as we talked about before. If you just put random variants in, random SNPs, this is for SNPs, then you'll get TITV of 1/2. So the closer you are to 1/2, the more random SNPs you have. Random SNPs are wrong. So Indel ratio is hard to measure. You need to distinguish between common and rare, or sometimes we use known and unknown for proxy for common and rare. In common, we have around a ratio of 1, and in rare, it's more like 0.2 or 0.5, between 0.2 and 0.5. So it's a little hard to know whether the variants that you're using are common or rare when you're working, for example, with a Finnish population. Which of those cutoffs do you use, right? Okay, genotype concordance is the concordance you want to use in a, here's how it's defined. So if you have your call set as the yellow and some other call set, which we call the gold standard in purple, you can count the number of overlaps. Stars, triangles are the ones that are false positives relative to the gold standard. And of course, the circles are false negatives that we didn't find. And so we can just ask what our sensitivity is. So we count how many true positives divided by how many true positives plus false negatives. We get 70% in this example. And false discovery rate, in this case, is 42%. We can also calculate the genotype concordance, which is just the matches divided by the matches when we talk about genotypes. Because there's a difference between comparing variants and comparing genotypes. But so here, that's going to be 4/7. So what do we use this metrics for? Again, we compare the concordance that we got. We can look at the concordance that we get versus concordance that other people got on similar projects to know whether this is a reasonable concordance. How do we calculate all these metrics? There's several different options here. I'm not going to read all the parameters, but both GATK and Picard have tools for both variant level evaluation, so things that talk about the variant set as a whole, and genotype concordance when we're comparing our call set with a truth to find out what the concordance is of the variant. You can go over these on your own. They're a little bit different. The main difference is that the GATK version, the variant eval, has more options for chopping up the data into parts. For example, you can distinguish between known and novel. What is the TVTV for the known? What's the TVTV for the novel? Stuff like that. With Picard, you cannot do all these things, but on the other hand, Picard is a lot faster, so it depends on what you need to do. In genotype concordance, Picard has an option of specifying that in a certain region, if it's not in the call set, it's not in the truth set, then it's definitely a homozygote reference. Right? So this is for cases, for example, if you want to work with the genomes in a bottle, in addition to giving you the VCF, they also give you an interval list, and they say, if it's not in the VCF, but it is in the interval list, then it's definitely home ref. Right? They promise. Outside that interval list, they make no promises. Right? So you can use that to calculate genotype concordance more accurately over that interval, confident interval with Picard. On the other hand, GATK has an option of, if you're working with very large call sets, has an option to parallelize this pipeline. So you call it on separate parts of the genome, and then you can merge the results together. So, all right. That's what I just said. Great. Okay. So in the end of the day, you calculate all these metrics, and you bring them to the group meeting, and you talk about them. And you do that in a very open way, because there's no way that at least we are not aware of a way of automatically answering the question, is this call set good or bad? And so you have to bring the metrics to a room and argue with your fellow researchers about whether you did the filtering correctly or not, about whether you looked at the right metrics or not, about whether everything looks good, right? So that's, in the end of the day, that's how it's going to be. And they'll send you back to the computer, and they'll say, "Yeah, you didn't calculate this metric, you didn't calculate the other metric, you should have excluded that sample and that sample, whatever." And then after a few iterations of that, then you'll decide to publish that paper. All right. Thank you.